Okay, let's see. We got eons, eras, periods, and then the next one is, yeah, epochs. All right. We didn't get the periods. All right. The point is, eons are long periods of time. Eras are slightly shorter periods of time. Periods are shorter than that even. And then finally, epochs. Are super short periods of time, just like 1 or 2 million years, if that. Eons are like billions of years, eras are like tens or hundreds of millions of years, periods are millions and millions of years, epochs are sometimes less. The point is, there's just this layer of length of time that we can break these things in. All right, we're going to talk about fossils. How does a fossil form? Being crushed around in the earth.
After millions of years.
But, okay, so let's back up. What has to happen first?
The animal has to die.
Okay, so the animal dies, and then what?
It gets like, it can be eaten.
The problem is, if it gets eaten, it gets digested.
Yeah.
And that probably won't leave a fossil. So it's actually relatively rare for an animal to become a fossil. It needs to die but not be eaten.
It has to be like covered by sediment.
And it has to be covered by sediment. And it has to be covered by sediment quick enough that it doesn't actually dissolve or get broken down by microbes, that sort of thing. So fossils, you often find them in what type of rock? Yeah, sedimentary rock, right? So 99% of the time you'll find a fossil in sedimentary rock. And they form in a couple different ways. Like, so for example, if you have like I don't know, like a shell here. There we go, a little shell. You can have a shell that gets buried and the calcium carbonate from the shell dissolves away. And then all of a sudden you have this shell-shaped hole in the rock, in the sediment, right?
I actually have one of those, like, of those shell in my house.
Oh, really?
Yeah.
Oh, that's cool. So you have this mold that's formed by a void, right? But then sometimes in the rock, in the void, there's these little cracks, and in the cracks comes water, and water starts to fill up or flow through, but then it deposits minerals, or crystals can start to grow inside here, and it will actually fill in that void. And so you end up with like a little crystalline shape of a snail shell. It's called a cast. So molds and casts are kind of the same type of fossil. But it's where the material has left that void, and then the cast is where it's been filled in.
Mr. Marcotte.
Yeah.
My brain is, like, I know what type of animal that was, but I can't, like, actually.
Like a nautilus?
Yeah, it starts with an A, I think. I'm not sure.
An amosite?
Yeah, I don't know.
You could also have another type of fossil called a replacement. Yeah, replacement. We can do better than that. So replacement, same sort of deal. You'll have like a bone or something. And then the bone tissue itself, instead of just completely molding or like disappearing or dissolving and then leaving a void, as it's sitting there underground in the layers, again, usually it's from water slowly dripping through and replacing it bit by bit with stone. So it'll start to get like more and more minerals until eventually the entire thing is just stone. Right? When you see like dinosaur skeletons, that's what's happened. Is usually the bone has been slowly replaced by a stone.
Ms. Margaret, when you mean eaten, do you mean digested or?
Yeah, I mean, in some cases, like an animal might be like partly eaten, but the bones aren't digested. But
Yeah.
if bones are eaten, the bones themselves will often dissolve in like stomach acid and stuff. So, or you know, like wolves will like break open bones and suck the marrow out, and then the bones are like scattered in chunks. And I mean, yeah, they might still make fossils, but
So it's like when they're like scavenger birds, like after like eat off like all the flesh, but then just leave the bones there.
yeah, but again, they'll scatter it, and so it doesn't make for a nice like informative fossil. You might just have a single bone or something like that for the fossil. But if you're looking for a whole organism, it definitely can't be eaten. Okay, any other types of fossils that you guys know about? You can have trace fossils. Trace fossils apply to not only things like coprolites, but things like footprints, right? Where like an animal comes along, leaves a footprint. You can have fossils in amber. This is like the classic Jurassic Park one, which incidentally isn't real because the video just talked about it. Like DNA doesn't really last past 1 million years. But mammoths, maybe. Dinosaurs, no.
Bro, bring it back to dinosaurs? How could that go wrong?
But there might be like little tiny fragments of DNA. And then finally, the last one is original material.
How does like plants get fossilized?
Same sort of deal.
Oh.
Often through like replacement or a mold cast. Usually less trace fossils. Original material, we have an example of this at the museum. Have any of you guys seen Babe the Blue Ox?
Yes. Yeah.
Yeah, he was just preserved in permafrost, right? He was like frozen for, you know, I don't know, whatever it was, 50,000 years, something like that.
Is it really dead though?
The ox?
Yes.
Yeah. Long.
But it's the original material, right? Like legs are bent up under him.
Oh, is that the thing at UAF?
The dude at UAF.
I'm pretty sure there's like, they have one there.
Yeah, exactly.
I like that Mr. Markov
That's-
says he's not a good drawer sometimes, but he just like duds that.
That's not a very good like.
That's good, Mr. Markov.
Okay, thank
I'm
you.
not
I
even
appreciate your I'll take the compliment.
Take the
Um,
compliment.
Yeah. Anyway, so have you guys heard of the La Brea tar pits?
Yes.
Yeah, same sort of deal. It's in California, right? Big tar pit. Animals like wander along, get stuck in them, can't get out. They like fall in the tar. It doesn't decompose. It just like preserves their bodies. So in the Arctic or in some of those unique situations, the actual bodies can be
Mr.
preserved.
Markov, have you seen the La Brea show?
I've heard of it. It doesn't really have much to do with the tar
I
pit.
know.
But it is, yeah, I did.
I like the first thing my head went to weirdly.
When I said La Brea, yeah, that's
Yeah.
fair.
I
Okay.
also went to the tar pit, so
How do you know how old fossils are? Well, we're going to do some dating.
yeah, carbon dating.
Yeah, we're going to do two types. Well, first we'll do relative dating.
You can't really see.
Mr. Markov's hand is in the way.
So relative dating is one of those kind of like no duh style moments, but it's worth mentioning that when you have like you have these layers in the stone, right?
You did see really.
Wow. Anyway, as you go back in time, you go through the deeper layers, right? So the oldest layers are at the bottom, the newest layers at the top. Not always true, you know. In rare cases, you can have like sections of the earth that over time are like uplifted and like flop over. But you know, 99.9% of the time, the newest layers of rock are at the top, the oldest layers of rock are at the bottom. Therefore, if you find a fossil above another fossil, The newest one is the one on top, right? So you can be relative. Also, sometimes in like geology, you can find like a layer. They don't even have to be in the same spot. You can find like this common layer. And if this layer appears also in a continent over, 1,000 miles away, you can say, oh, well, this other organism I found is above this layer. This other organism is right below the layer. Therefore, this organism occurred after. I'm thinking of like the KT boundary, or like he was talking about in the video. They mentioned some of those layers as well.
Yeah, and video that we watched Wednesday.
Yep.
That's a good video.
And then the other one you talked about was carbon dating, which is a type of radiometric dating. So in radiometric dating, you have these radioactive isotopes in the decay. The one in the video they mentioned was uranium. So U238. This is an isotope of uranium. It refers to how many neutrons are in the uranium. And over time, because this isn't stable, it decays, it falls apart into a bunch of chunks. It talked about some of the steps, but we're just going to skip to the end. It becomes lead. PB206. How long does it take? Billions, actually, on this one. So this one's about 2 billion years old, or 2 billion years. And specifically, this is a half-life. So that means over the course of 2 billion years, not all of their uranium will turn into lead. It's not like, bam, 2 billion years are up, and then all of a sudden it just turns. What it means is, after 2 billion years, half of the uranium-238 isotopes that are out there will have turned into lead. And then 2 billion years later, half of those will have turned into lead. And then 2 billion years later, half of those will have turned into lead, right? So uranium is a good isotope to use when you're trying to calculate the age of the Earth.
How do they carbon date stuff? I'm curious.
We'll do carbon next, yeah. So carbon is the isotope carbon-14. Carbon-14 decays into nitrogen-14. And this guy takes about, it's like 5,000, I'll put 6,000, I think rounds up. Approximately 6,000 years is its half-life. The amount of carbon-14 in the atmosphere is relatively constant because carbon-14 is made basically by sunlight, like striking carbon, or like it's just made basically using UV light from the sun. So the carbon-14 in the atmosphere is just hanging around at a pretty constant level, which means that the carbon-14 in living plants is pretty constant, right? Because like the plants take in the carbon-14, which means the food we eat has carbon-14, which means your body has carbon-14 that's pretty similar to the atmosphere, right?
No.
What happens after you die?
All the carbon like releases your body.
Well, the carbon doesn't, you don't bring new carbon in your body, right? So all of the carbon in your bones and stuff like that is no longer being replaced by the carbon in the atmosphere. So therefore, the amount of carbon-14 will go down. And it goes down at the rate of half of it turning into nitrogen every 6,000 years. So if you were to find a bone, if like anthropologists go out there and they find like a human bone or even animal bones, something like that, but if it still has carbon in it, it hasn't been totally fossilized, and if they're like, oh, look at this, it has a quarter of the carbon-14, 25% of the carbon-14 is remaining in it, how old is it?
I want to say like 18,000 years.
Is it 3,000 years old?
1,600 years.
1,500 years?
I'm guessing like 600 years. No.
When it died, it had 100% of its carbon-14, right?
9,000.
And now it's down to a quarter of its carbon-14.
9,000. No, 12,000. No.
2,000.
I'm guessing 12.
But we're going the other way. So it's actually 12,000 years.
I got it. After like
After like
much
80 guesses.
struggle.
You've made a lot of wrong guesses.
After like 20 years.
You guys see why it's 12,000 years though? The first 6,000 years, you go from 100% of the carbon-14 to what? Half, right? So if we do a little timeline here, this is 6,000 years. You go from 100% down to 50%, right? And then 6,000 years later, you go from 50% down to 25%, yeah. So the smaller the fraction of carbon-14, the greater the age.
Mr. Marco, I had the correct answer in my head. I was like, okay, it's half.
This is a great test question, by the way. Hint, hint. Is to do something really similar to that. This is carbon dating.
My head was struggling on that. Way
Yeah.
too much.
It doesn't work since 1945. Why?
Because after that would be too long.
Well, we started exploding nuclear bombs in the atmosphere, and the amount of carbon-14 just like exploded. Like a nuclear explosion creates tons of carbon-14. So now the scale's all wonky and weird. So it's really hard to do things since the 40s. But anything older than that, carbon-14 is pretty accurate. But of course, there's a limit. If you're down to like one tiny little like thousandth of the original amount, the accuracy is really poor, right? So carbon dating is only effective down to like 50,000 years ago or so, something like that. Okay. Let's talk about what's left. Oh, origins of life.
Oh.
So back in the day, before they knew about microscopes, they were like, hey, look at this. I take a piece of meat, I let it sit on the counter, and all of a sudden, maggots just appear out of nowhere. Whoa.
Sorry.
This was called spontaneous, knew someone was going to say that, generation. They used to think like, oh, life can just appear out of nowhere, right? Because they didn't really understand like where those maggots were coming from. We know where do the maggots come from? Flies, yeah. A fly zips along, lands on it, drops the maggot, flies away, right? It drops off some eggs. Same deal with like bacteria and stuff, right? We can see mold. Mold can just appear on bread. Poof. Where did the mold come from?
Magic.
Magic? Out of nowhere? Did it
Yeah.
just like...
Came from a moisture.
It doesn't just appear from nowhere. Where does the mold come from? Mold spores, right? Floating around in there. There's just mold spores floating around just right now in the air. Same sort of deal. So we know this isn't really a thing, right? We've most of you guys are pretty aware of that. Back in the day, they didn't know about that. But they started to disprove it. And one of the simple ways we can disprove it is we can make some delicious canned salmon. All right?
That's the best.
Yeah, it's pretty delicious. So you take your meat, you stick it in a can, you seal it up, right? And then you sterilize it. So you apply heat, it sterilizes inside. As long as it stays sealed, does it go bad?
No.
Do things grow inside it? No, if you do it right, nothing grows inside it, right? It stays good. And then people are like, oh yeah, that's weird though. That just means it doesn't have any oxygen. So therefore, obviously, things can't just appear. It's not exposed to the air.
There's still oxygen in there.
I mean, yeah, there is, but there's not fresh oxygen.
Yeah.
Anyway, so this one guy, you may have heard of him before. I try not to name drop too much, but this one's worth it. His name is Louis Pasteur. You've heard of like pasteurized milk? Right? Yeah, you can thank this guy.
Okay.
He did a similar sort of thing where he sterilizes like a nutrient broth, but this time he leaves it open to the atmosphere, but the container he used had this big like long swan neck. So he had this nutrient broth. So it was open to the air kind of, what happened was the little like mold spores and the bacteria that are floating around in the air, they'd float down this neck, but then they'd settle right about here. And they couldn't make it up this part and back down. So his sterilized broth, even though it was open to the air, remained sterile. So he was proving basically that things go bad not because of spontaneous generation, but because cells, as we know, or like spores or whatever, drop into it from the air. So the other question about the origin of life. So then going back, we know that spontaneous generation doesn't happen. This is part of cell theory. We've talked about it already a bit. Where did the first cell then come from? How did that actually evolve? And like what was possible there? Nobody really knows for sure. We'll talk about it in a sec. But some of the building blocks to make that cell, people thought, oh, well, where do those come from? And it's hard to know because here on Earth, everything has been contaminated by life. Everywhere we go on Earth, we find life. They dig like under the ice shelf in Antarctica, right? Like miles down, they find things living down there. Drilling down, like when they drill oil wells and stuff, all the way down, there's still life. There's still microbes living all the way down as far as we drill. Up in the atmosphere where the airplanes fly, they find microbes living up there. Everywhere has life. Everywhere has been contaminated with organic molecules like amino acids, stuff like
But
that.
there's life in space.
Um,
That's the question.
So, yeah, we haven't found any there outside of Earth.
Actually, on Mars. Did we find some on Mars?
There are materials like the basic building blocks, those the atoms for life are present on other planets, right? So things like ammonia, NH4, things like carbon dioxide, or methane, actually be more appropriate. I'll change that to methane. Here. CH4. Water is on other planets, H2O. Even Mars has water, it's just not liquid, right?
Yes, bro.
And so these guys, Miller and Urey, they devised an experiment. They're like, hey, using these sort of like inorganic molecules, can we make organic molecules? And they theorized that it was possible. And so they, again, built an apparatus. So they took this tank full of these molecules and they heated it up. And then they brought it over to this other chamber. And in this other chamber, they electrified it. So they had this electrode. Gives like a little spark between them. Look, something like this. Boom, boom, and there's a little spigot here. They put a beaker. And they took these inorganic molecules, the ammonia, the methane, the water. They theorized this is like the primordial soup of ancient Earth, right? And they heated it up, and then they zapped it with electricity. The idea being that if you get like energy from lightning strikes or volcanoes or radiation or whatever, that energy can change the molecules and reform the bonds in different ways. And it does it kind of randomly, but if it happens enough, and so they cycled it through quite a bit, and they found they could collect things like amino acids, nucleic acids, etc. They didn't build DNA from scratch, right? It's not like actual things, the polymers, but they were able to create some of, and not even all of them, the building blocks of life. Yeah.
Wow.
So this was kind of part of the like theory of how organic molecules. They proved that at least organic molecules could, in theory, come from inorganic sources. We aren't able to test this empirically because we haven't found these building blocks on like other planets, and we can't. Anywhere we look on Earth, we're going to find these things because they're just strewn all over the place. Nobody's really sure on like the origins of life as far as like how life arises. But some of the things that need to happen is you need to have like DNA that somehow, or RNA that somehow replicates itself. Again, there's theories out there. None of them have particularly strong evidence one way or another. You also need to have membranes of some kind. There's a theory that like the membranes contain nutrients that allow this DNA to do its replication. And then somehow the DNA that can replicate ends up inside of these things. Again,
Not proven.
no one, yeah, no. I mean, proven is kind of
Harsh word.
a too strong of a word for anything in science, but there's no particular evidence to support all these competing theories. But all we really need to do to call it life is to have DNA inside a membrane and then have that replicate. And then from there, it's kind of able to evolve. But how that first situation arose, nobody's quite sure.
Time travel.
Time travel. Okay, but remember, this is actually kind of, in theory, the easy part, because this only took 500 million years. The part that took 1.5 billion years was the making of eukaryotic life. So it's worth mentioning that one. Here's the theory behind this. And this one does have really good support. As far as we can tell, this is exactly what happened. There was a bacteria, a heterotrophic bacteria, and the heterotrophic bacteria basically takes like chemical energy from things around it, right? Like if there's sugars around, it can take those sugars and then turn it into ATP and go about its little life. That's what it means to be a heterotroph, right? Anyway, along came this archaean, and the archaea was bigger, and what it likes to do is it likes to eat heterotrophic bacteria.
Yum, yum.
So
It's Pac-Man.
it comes along, it goes to engulf him, right? That's how cells eat each other.
You ever seen Pac-Man with rail?
Okay, so inside, it now has this heterotrophic bacteria, but something happened where instead of digesting it and consuming it, it figured out, oh, wait a minute, if I give this guy sugar, he'll give me back ATP. So they formed a symbiotic relationship, right? Not the nucleus that I'm going for with this one. What organelle does this guy become? Yeah, this becomes, over time, mitochondria. And then it happened again in some lineages. With a photosynthetic bacteria. Same deal. Came along, went, oh, look at that, yummy. Went to eat it. It found, though, that it's. I'm going to put the mitochondria in there, too. It might be more beneficial, instead of eating the photosynthetic bacteria, to just keep that photosynthetic bacteria inside you. And it can make all of the carbon molecules, the sugar, from the sunlight instead. This becomes like a plant cell with mitochondria and chloroplasts. This whole thing is called endosymbiotic theory. endo being like inside of, symbiotic, again, because they started out with like the symbiotic relationship where it was a completely separate organism, but then eventually became just a part of the larger organism. And we have three main pieces of evidence for this symbiotic theory. Does anyone know any of them? How are these organelles like bacteria? They can't, but they do some things on their own. What do they have on their own?
They have the ability to make ATP and stuff like that on their own.
Yes, kind of. Just curious if anyone knows any of these other ones. What else can they do on their own? What happens when you're like, for example, a muscle cell, you need lots of mitochondria in a muscle cell, right? How does the muscle cell make more mitochondria? Does it make them from scratch? No, you don't consume more bacteria. What was it? It builds mitochondria from scratch? Oh, does anyone know how, if your body needs another mitochondria, if it has one and it's like, oh, I need 2 mitochondria, how does it make another mitochondria? What was that? Yeah, it splits. The mitochondria, the cell somehow can tell it to grow bigger and then split in half. Boop. That's binary fission, right? They both undergo binary fission. Bacteria, same way bacteria reproduce, right? Get bigger, split in half, boom, done. Also, did you know that mitochondria and chloroplasts, they have their own DNA? They do. The mitochondrial genome is about 16,000 base pairs. You inherit your mitochondria from your mom, so you get all of your mitochondrial DNA from your mom. And then finally, this one's a little bit more subtle. I drew a extra membrane around it, so when a cell engulfs another cell, it surrounds it in its own membrane, right? But the cell that was just engulfed has its membrane. Both mitochondria and chloroplasts have a double membrane. So these are the three, I think, strongest pieces of evidence. Plus the DNA that's inside a chloroplast matches up pretty well with the photosynthetic bacteria. Chloroplasts have lost some of their genes to their host cell, so they're completely dependent on that host cell. It's no longer a symbiotic relationship. They're just part of the host cell. But it's kind of cool to think about. All right, we're going to stop there on the notes. Thanks for being patient with that.